today's review video, we will be going over photoreceptor cells. There are two cell types when in reference to photoreceptor cells. We have the rods and the cones, as demonstrated in the figure presented here. Rods and cones are very similar in their general structure, as they both have a synaptic terminal, which contains synaptic vesicles. They both contain an inner segment that contains a nucleus and mitochondria, as well as a similar but slightly different outer segment, which contains plasma membrane for both, while the rod has the cytoplastic space and disc for additional uses and additional visual aids. For the rods are responsible for monochromatic vision, night scopic vision, as well as there's almost 130 million rods presented in the human eye. Cones are responsible for color, as well as day phototic vision, and there are almost 6.5 million cones, as the cones are slightly more sensitive to that of the rod cells. There's the question of why we have two photoreceptor cells, and the reason is that a single type of receptor cannot produce both high sensitivity and high resolution. So this requires that we have two separate cells for these functions. As previously explained, the rod cells and cone cells are the two that humans have. The rod cells are responsible for the high resolution. This can be due to different methods of being able to break down the light that's entering the retina. This can be done through massive convergence, which is when there is a large amount of rods connected to a smaller amount of bipolar cells to then one ganglion cell that relates to the optic nerve fiber, while on the other hand, the cones have a more one-to-one direct relationship with one cone going to one bipolar cell, to one ganglion cell, and then to separate optic nerve fibers. This allows for both high sensitivity to be determined by the cones, as it's a one-to-one relationship, as well as high resolution to be made by the rods.